Hey everyone, I'm Emma, and this is my experiment on teratogenic substances and living organisms. So what are teratogens? Teratogens are toxic substances that affect the fetus during pregnancy. I chose to do this experiment in order to better understand the effects of teratogens on living organisms. We know that teratogens have their effects on pregnant mothers and their children, but I also wanted to know if there were similar impacts on plant life. So the hypothesis being tested in this experiment is that 4.5 alcohol, 9.9 alcohol, vitamin A supplements, and nicotine, which are all teratogenic substances, would have a negative impact on mung bean plants. I also predicted that the 9.9% alcohol and the nicotine would likely slow the growth of the bean plants the most. During the experiment, the materials used were mung bean plants, tap water, 4.5 alcohol beer, 9.9 alcohol beer, cigarettes, vitamin A supplements, paper towels, paper cups, paper plates, measuring spoons, a ruler with millimeters, and environments with equal conditions. The steps taken were placing 9 mung beans on a plate with a paper towel and labeling the 5 plates with tap water, which is the control group, and then our independent variables, the 2 types of beer, nicotine, and vitamin A. Then, each day I would pour 1 tablespoon of each of the teratogenic substances in the control group onto the mung bean plants and observe them for growth and changes. After 6 days, the experiment ended and the results were in. We found that water, our control group, had a final average length of 6.2 millimeters. 9.9 alcohol beer had no growth at all. 4.5 alcohol beer had a final average length of 0.6 millimeters. Vitamin A had a final average of 2.7 millimeters. And nicotine had a final average of 1.8 millimeters. So what does this mean? This meant that the hypothesis that the teratogenic substances would have a negative impact on plant growth is correct. Water, our control group, still grew the most and the fastest out of all 5 substances, while the teratogenic substances grew very slowly and at a very minimal length. Despite the clear results of the experiment, there are some confounding variables that we need to consider for next time that we do the experiment. Some of the variables that we need to consider are equal ratios, especially considering the dissolved vitamin A and nicotine in the water, and the freshness and potency of the substances, especially regarding the beer, which typically goes stale after 1 day. We may also need to do more research in the future, especially regarding how teratogens might affect other living beings and the specific chemical impacts that they have on developing fetuses. All in all, the experiment was successful and showed that teratogens do indeed have a negative impact on plant growth and the development of living beings. Thank you guys for taking the time to check my experiment out, and I will see you next time. Bye!